So far, we've been treating ventilation and perfusion as just the flow of two fluids, water and blood, with not much attention paid to the nature of these fluids. We spent some detail on flows on the ventilation side, that is, how water is pumped over the gill surface, what direction it flows, and so forth. But what about on the perfusion side? There are some pretty significant adaptations there as well. We'll start with the tale of two fish, two types of fish actually. On the one hand, we have nearly all fish in the world which pump nice red blood through their gills and bodies. On the other, we have these fish. This is a representative of a family of perciform fish, the Conichthiidae, also known as ice fish. There are some 11 genera in this family, and they all have distributions in the Southern Ocean, mostly the very cold waters of the continental shelf around Antarctica. Their name doesn't come from the icy waters in which they live, though. The name comes from their translucent bodies and from an unusual characteristic. Unlike nearly every other red-blooded fish in the world, the blood of the ice fish is clear and yellowish. What accounts for this difference is the nearly complete absence of the pigment hemoglobin that tints the blood red of every other vertebrate in the world. Hemoglobin is one of a broad class of metal-containing proteins, or metalloproteins, known as respiratory pigments. We'll have more to say about respiratory pigments in general a little bit later, but let's focus for now on hemoglobin. Hemoglobin aids in respiration by acting as an oxygen sponge. Among vertebrates, each hemoglobin molecule can absorb 4 molecules of oxygen from oxygen dissolved in water. We say that it can act as an oxygen sponge because the binding of oxygen to hemoglobin is easily reversible, releasing oxygen back into soluble form again in water. The physiological significance of this is that hemoglobin can considerably increase the oxygen transport capacity of the blood. Put in terms of the Fick principle, the presence of hemoglobin has the effect of increasing the solubility of oxygen in blood. We're not really increasing the solubility, of course. The Bunsen solubility is a property of water that is independent of whatever hemoglobin is doing. Rather, we're adding an additional storage reservoir for oxygen in blood. Conventionally, this is expressed by adding a term, beta, to the solubility term, but that's a long equation, so we're going to lump those together in what we will call an apparent solubility. Irrespective of how we define solubility, however, the overall effect is obvious. Increasing the apparent solubility of oxygen in blood has the effect of increasing the oxygen transport capacity of the blood. This means, for example, that blood can transport more oxygen for the same volume flow rate or for the same arterial venous partial pressure difference compared to when hemoglobin is absent. The effect can be considerable, which we can demonstrate with a simple calculation. The molar concentration of hemoglobin in blood varies quite a bit between animals and depending upon condition, but a rough rule of thumb puts hemoglobin concentrations typically at around 2 millimolar. When the hemoglobin is fully loaded up with oxygen, that is, when each hemoglobin molecule is carrying its full capacity of 4 oxygen molecules, the molar concentration of oxygen-bound hemoglobin can be as high as 8 millimolar. Now, let's remember how much oxygen can be dissolved physically in water. We just go back to Henry's law, do a calculation using the Bunsen solubility of oxygen in water at, say, 20 degrees. This is about 1.3 times 10 to the minus third millimolar per liter per atmosphere. And with a typical atmospheric mole fraction for oxygen of about 0.21, we do the calculation and we see that the maximum concentration of dissolved oxygen is about 0.275 millimolar. We can now see the extent to which hemoglobin can increase blood's carrying capacity for oxygen. 8 millimeter on hemoglobin over 0.275 millimolar in water, and we see that the hemoglobin has increased the carrying capacity of blood for oxygen by a factor of about 30. Turning back now to the Fick principle, we can see that blood-containing hemoglobin can transport 30 times more oxygen for a given volume flow rate of blood. The most interesting consequence of this can be seen with the ventilation-perfusion ratio. Just as a reminder, here it is again in its long form. If we take a look at what this should be for the Antarctic ice fish, we take into account that both the ventilated and perfused fluids are mostly water. Their Bunsen solubility coefficients should therefore be about equal, which means that the ratio of their solubilities is going to be about 1 to 1. We've sort of been implying this was the case all along, but if we turn to every other fish in the world which has hemoglobin in their blood, we see how wrong this assumption has been. The presence of hemoglobin at typical concentrations pushes this ratio of solubilities to a value considerably less than 1. By our calculations, it's about 1 over 30. Let's now remember the inexorable rule for the ventilation-perfusion ratio. This formula has to always equal 1. Thus, we see some interesting consequences of this permanent change in the solubility ratio. Either the ratio of volume flow rates has to be 30 over 1, or the ratio of the partial pressure differences has to be 30 over 1, or some combination of the two together has to equal 30 over 1. This is an ironclad rule. Remember, the ventilation-perfusion ratio is a consequence of the unbreakable law of conservation of mass. It's not optional. We can see the consequence of this in some of the adaptations of Antarctic ice fish compared to normal fish. So, for example, one of the ways to increase the volume flow rate ratio is to decrease the perfusion rate of blood to the gills. So, fish with hemoglobin in their blood should have generally lower perfusion rates than do Antarctic ice fish. In fact, this is the case. Antarctic ice fish have hearts that are roughly 4 times larger than that of other fish, and they have very wide blood vessels throughout and very high capillary densities throughout. All of these adaptations aid in supporting an extraordinarily large output of blood from the heart. Indeed, this can be a problem for ice fish because the pumping of the blood at these high rates can be energetically very expensive for them compared to the pumping costs for other fish. We also see another trend in fish evolution explained by hemoglobin's persistent downward biasing of the solubility ratio. We mentioned several times so far that the fish gill is an extremely efficient gas exchanger. This means that the fish gill is capable of extracting a large proportion of oxygen from the ventilated stream, in other words, maximizing the delta P in the ventilated stream. And this is precisely what we see. Hemoglobin was a revolutionary development in the ability of vertebrates to efficiently transport oxygen in their blood. And in the case of fish, as we've seen, it has shaped not only the energetics of circulation, but the design of gas exchangers as well. It's no coincidence that the countercurrent exchange system of the fish gill is one of the most efficient gas exchangers in the animal kingdom. So let's take a deeper look at these remarkable pigments.